So we have another tool in our arsenal, antimicrobial phages, AMPs. They are short-chained amino acids, usually ranging from 12 amino acids to 50 amino acids, and they usually have a positive charge and show partially hydrophobic nature. They are used to kill these bacteria. Antimicrobial phages usually act on the membrane, and they show three types of targeting mechanisms. First is barrel targeting, in which the pores are injected in the membrane and barrel-like pores are formed on the membrane which break the membrane further. And second is the toroid pore model, in which the phage changes the topology of membrane and breaks it further. And third is carpet model, in which the phage binds around the membrane and then it dissolves the membrane inside it and killing the bacteria. Further, and AMP action can be done inside the cell as well. Some of the examples for that are lysozyme, which is present in our saliva and in our tears, which kills the bacteria. Another example is niacin. It is used to prevent food from rotting and is also used in clinical purposes for some reactions. Third is deferensins. Deferensins is for punching pores inside the membrane. And fourth one, cathelicidin. It kills the bacteria and also guides the immune cells for further actions.
Causes of AMR. Resistance doesn't just apply randomly. Our behavior accelerates it. Overuse. Taking antibiotics for viral infections like colds or flu, which is not a good thing, because colds and flu are not always the things which are used for antibiotics. Misuse. Stopping treatment midway while the symptoms are improving. Agriculture. In many countries, livelihood and livestock are routinely given for antibiotics to promote the growth, which is a completely wrong thing. Poor infection control. In hospitals and communities, resistant bacteria spreads easily when hygiene lapses. To fight this back, we can do proper things such as using antibiotics only when a certified doctor is prescribing it, complete the full course of the treatment, prevent infections with hygiene, sanitation and vaccination. Support innovation. We can try to create new antibiotics with some more alternatives like bacteriophages and antimicrobial peptides. Remember, every dose matters and every action counts. Bacteriophage. One promising alternative in the bacteriophage is that it's literally a bacteria eater. Phages are viruses that infect and kill the bacteria. These are the most abundant entities on the planet. These are available in the soil, even in the water, even in our bodies. Well, there's a classical example of this bacteriophage, which is T4 bacteriophage. It looks like a microscopic spaceship, although it's not. It has a icosahedral head with DNA, a collar and a tail sheet, and a base plate with filters for attachment.
Imagine a world where a simple cut, sore throat, a minor infection could be deadly. This is not science fiction. This is the looming reality of antimicrobial resistance, AMR. Developed in early 20th century, antibiotics revolutionized the world. They treated the once fatal diseases curable. Developed in early 20th century, antibiotics revolutionized the world. They made the once fatal diseases curable, enabled safe surgeries, and saved millions of people. But today, microbes evolve faster than we could develop new drugs. Antimicrobial resistance happens when bacteria, virus, fungi, parasite doesn't respond to the medicine that we develop for them. Every time we take antibiotic, most bacteria die. But some of them have special survival genes that made them alive. They multiply and pass on this ability to the further generation, which make the diseases once curable but now difficult or even impossible to cure. Already there are millions of deaths happening due to AMR these days. By 2050, it can reach to 10 million lives lost due to AMR annually.
Well, we'll be talking about lytic cell step by step now. Adsorption. Tail fibers recognize bacterial receptors and land on the surface. Penetration. The tail contracts like a syringe, injecting a viral DNA. Synthesis. Bacterial DNA is degraded, phage DNA takes over, and directing protein and DNA synthesis happens here. Assembly. New phage heads and tails form and join. This is the most important part of the adsorption and even the steps which are required for lytic cycle. Release. The cell bursts, releasing hundreds of new phages. And now comes the lysogenic cycle. In the lysogenic cycle, the phage DNA integrates into the bacteria genome as a prophage. It multiplies silently with the host until it triggers the host. For example, by using the UV light, we can enter into the lytic cycle, which will influence the lysogenic cycle too.
Antimicrobial resistance is like one of the biggest challenges in our time, but we are not helpless. Through antibiotics, bacteriophages, and antimicrobial peptides, we can protect the future and fight stronger, smarter, and safely against tuberculosis.